So yeah, Brandon Ring is my name. Thank you firstly to Heidi and Callie for the opportunity to speak here today. So looking forward to it. So we've heard quite a bit about the interesting use of genomics and actually the previous speakers, Laurent and Lucas, but also from some of the other speakers yesterday. So in this talk, I'll be covering how genomics can be used in the factory environment to enable a more predictive and consequently a more preventative approach to food safety. So we'll look at a couple of case studies on this and as well as we move down through it. So all right. So just to start off with quite a startling statistic from the World Health Organization. Over 400,000 deaths annually from foodborne illness. So that's really quite shocking of a statistic. So something needs to change, and obviously the current systems just aren't good enough if this is continuing to happen. And this is something that industry as a whole is working to try and improve. So, but luckily, there is actually change happening, and these types of changes we can use to our benefit. There's so many changes happening right across the industry. So, a big one being data analytics and AI. Massive changes in computing power that we can tap into now. That was never available before. So this we can really use this and exploit this to our benefit. And IIoT, the Industrial Internet of Things. So there's been a massive shift in the availability of sensing devices and the technology around this. So it's now easier than ever to capture information, capture data right across your factory or from weather data, wherever it might be, and feed that in. So that there's been a real revolution there. And of course, genomics, as we've been talking about earlier, and over the last few days. So massive change happening across the industry here. So we've seen the cost drop here. So it's now becoming much more affordable than it used to be. So Adrian from Nestle touched on this yesterday as well. So it's still a work in progress, I guess, but it's the change. It's all moving in the right direction, and something now that's actually accessible and usable, and where previously it wasn't. So I guess as we look at this, it's good to compare why is it important, or why should we embrace genomics. So really one of the challenges is that only about 10% of bacteria can be detected using traditional culture techniques, and essentially this is just the tip of the iceberg in terms of the bacteria that exists in factories and in food products. So we're not really getting the full insight as to what's happening. So next generation sequencing that solves this. So we can see the full microbiome. So it just brings a whole new level of insight that a factory manager can use. So much deeper insight can be gained. Other limitations of traditional techniques is it's typically one plate per organism. So it's just not practical to look at many bacteria. It's done one plate at a time. So it's not a scalable solution. So it leaves you essentially looking to try and find indicator organisms, and they tend to not be that accurate. So you're very much looking at a reactive response, and oftentimes it's too late. When you found it, it's already too late. So using NGS, that's where looking at, you can analyze the full microbiome and observe how it's changing over time or in response to stimuli like temperature or humidity. So this is kind of a step in the direction of prediction and essentially prevention. And essentially that's really what we'll focus on for the rest of the talk here. Okay, so the first thing we're going to look at is a couple of case studies, and the first one is actually going to be on some product. So we're going to see just an exploration on some cheese, actually. So you'll agree that these two cheese samples, they look the same to observe them. And actually, when they were tested using traditional culture techniques, they both came up the same as well. They both appeared to be the same. The results were the same from each type, but there was an issue. So they were not actually the same. So one of these cheese samples actually was forming blisters on the packaging, um, and it was suspected to be causing from some off-gassing. But the root cause couldn't be identified, and this is causing a lot of frustration with the company that we're working with. And they could not see the difference between them. So they asked us to have a look at it. And when we use genomics as a tool, it was very obvious difference between the two microbiome. So very easy to identify the particular bacteria that was causing the off-gassing, and implement a root cause. So these new tools, it's got almost like the invention of the microscope. Towards the naked eye, it reveals so much more information and that you couldn't see before. Okay, so we're going to look at some other case studies. Actually, this from factory environments. So this is part of a project we're involved in with Professor Shay Fanning from the University College of Dublin. So we analysed a microbiome of 5 factories over a 2-year period. So a very unique project, and actually came some really interesting insights. So we were doing the bioinformatics and analytics on the data, and providing the insight back to the company. Um, so just to look at some of the insight that we got from this. The first one here, just to look at a seasonal trend. So and just to understand this chart here. So across the x-axis you have time, and there's sampling periods over the 2-year period. And on the y-axis, it's what's called relative abundance. So essentially, each color here is a particular bacteria. Um, and we looked at more or less the top 15 bacteria, and then all others, maybe 1,000 others, just for clarity, is all put into this cyan color into so you can get to see the bacteria that's relevant. So you can see quite a nice cycle here. So for this one particular bacteria, and it influenced the microbiome of other bacteria as well. So where it is quite low in autumn, and increased in spring, and then repeated again and repeated again. And actually we saw this across a number of facilities. So so this gives an insight as to how you might address your sanitization or your cleaning steps, or the particular actions you may take for different seasons. And this is the factory microbiome we're talking about here, not even the product microbiome. So so not obvious that the factory microbiome would change so much with seasons. So another trend we saw was on weather, actually. So we actually, during this project, we had an unusually hot summer in Ireland. And for anyone who knows Ireland, you'll know that a hot summer is certainly an unusual event, not something we see here too often. But there was a spike in one particular bacteria, and this bacteria is known to be temperature sensitive. And we observed this across a number of facilities. So you can see how just in this one simple by one simple data set here, we can identify that weather and temperature plays an impact on the whole microbiome. Because when you just any part of any bacteria, it impacts all the rest of the microbiome. So you can see how feeding weather data into your analysis can bring so much more insight. Okay, and a spatial trend that we saw as well. So in terms of location, so you see on the diagram here, so you have a changing room, and then a control zone facility here. And each of the dots represent where we took some samples. So this bacteria that we're looking at here is related to human skin. And so you can see it's quite dark here in the changing room. And then as you propagate away from the changing room, you can see how the concentration of that bacteria changes and declines. So, a really good tool for validating gowning procedures or zoning control measures, or whatever you may have in place. So you have, again, insight that's not obvious using other techniques. Okay, so as we move forward into, I suppose that was kind of more analytics. We're getting into a little bit more complex techniques now, looking at clustering technologies. So this is a graphic that shows the microbiome of each plant. Um, and so these are the 5 different factories that we looked at. So they're colored by factory here. So, and essentially how these cluster plots work is that dots that are close together, the microbiome are very similar. And plots that are or dots that are far away, the microbiome is quite different. So, one of the big messages here is that there's an obvious difference between the microbiome in the factories. You can see how they're different, but they also overlap in some areas as well. So, but essentially, each factory has its own unique microbiome, and I know that food companies will be well familiar with this, where oftentimes an old factory can actually run along quite stably, and a new factory with everything pristine actually you can have a lot of challenges there with bacteria because it's not so stable. So yeah, so as we actually I've explained that one now. So so when we drill into this, and this is a similar type of a chart where we're looking at one particular factor here in this case, and in this case we're coloring the samples by date. So a different set of a different type of analysis here, but we can see that there's this anomaly over here on the left hand side. So we know something happened, something shifted the microbiome, and we know that this was actually midway through, and so we didn't reassuringly we know that the microbiome reset itself again by the time we got back to take the next round a couple of months later. But what caused it to shift? That's the critical question. So yeah, so so now we've recolored these by sample type, and this was touched on previously, but we can now see actually that this is a raw ingredient here, and you can see that it the particular bacteria in question here is something that is known to occur in the raw ingredient. And it was identified that actually there was a spill from this particular batch in the factory. All of these other samples here are swabs from the factory environment. So a batch came in that was contaminated with this particular bacteria. There was a spill, and it caused a shift in the whole microbiome on the factory. So really something that was quite startling to the company involved here, that you could have caused such an issue across such a wide part of the factory by this incident. So it really gives some really interesting insight. Okay, so I guess where is this all leading to? So I guess, so how do you actually pull all of this together, and how do you implement it in a factory to enable preventative food safety? So first step, always just getting some data in, and companies have so much data to hand already, and generally it can be often quite easy to access. So just plotting this alone can give you quite valuable insight. So that's really the first step to get onto this journey, and to start to capitalize on it. So then as you move forward, you can bring in some additional data sets. Obviously, we talked about genomics data. So, obviously bringing in culture data and weather data, we saw that they're quite easy. Genomics data, obviously a little bit more challenging, but again, far more insight to be gained from it. So condensation is another quite interesting parameter as well. It's actually quite complex in terms of when it occurs, and there's so many factors that influence and affect whether condensation will occur, and these can change by the hour, almost nearly by the minute. If you're doing a cold flush through a pipe, condensation can occur on it, and obviously moisture, water availability has a big impact on bacterial growth. So so this these are things that can cause a shift in your microbiome and increase the propensity for pathogens spoilers to occur. So yeah, so bringing all of that together in a dashboard, then you can, having it in one location, then it gives the factory managers a very simple interface to come and look and view and understand what's happening. Spot anomalies and trends happening over time. Drill into the data. Conduct root cause analysis to try and find out what's happening in the factory and really actually use the power of actually the human eye is actually quite powerful for spotting anomalies and trends as well. So next step in this journey is actually integrating all of this into a model, into predictive models, essentially algorithms that can analyze and process all of this data and other data sets as well. And this is really where you get a step change in prediction. So again, as we mentioned, the changes in industry that we can tap into in terms of computing power and machine learning that's now available to us all, IIoT, etc. So this is really where it all comes together and really starts to actually bring enormous value to the user. So you can analyze, visualize your process flow, see where there's issues happening, click into it and understand and observe trends over time, or trends by process, or trends by severity as to particular bacteria that you're most concerned about, pathogen spoilers, biofilms, etc. And be able to sample what site or what factory that you want to look at, and have recommendations built in as to how you can drive change and how you can implement and improve and see what's happening out over time. Okay, so that's pretty much what I wanted to cover. So just a recap on we know change is needed, but luckily we know change is happening, and as the type of change that we can all benefit from. And we see that prevention is possible. We're working on projects in this space with some very large companies, and they're seeing the benefits of the use of this technology. So it's really an exciting time. So very much now is the time essentially for industry to come together and really drive this change and really implement it. So with that, just to say thank you, and I'll hand back to Heidi to cover out from here. So thank you very much.